Hello everyone, welcome back to Agrivyas Academy YouTube channel. So in this session, we'll continue with the special chromosome. So now let's discuss about the second important special chromosome known as the giant chromosome. So before going into the topic, so let us know about the what is meant by the term giant. The term giant nothing but very large. The term giant nothing but very large or very big or huge. So the chromosome which are very large or very big are known as giant chromosome. So what are giant chromosome? By the definition, we can say as the chromosome which are present in certain tissues of some organism. These are specialized and abnormally these are very larger in size. Such chromosome are known as giant chromosome. That is, in in some tissues of some organism in some tissues of some organism the specialized abnormally larger chromosomes are present, and those chromosomes are called as giant chromosome. Those chromosomes are called as giant chromosomes. Those are called as giant chromosomes. So who discovered these giant chromosomes for the first time? For the first time, these giant chromosomes were discovered by E.J. Balbiani. E.J. Balbiani in the year of 1881. Later, these chromosomes described by later described by these were described or who explained the significance of giant chromosomes is painter is painter in the year of 1930. So the giant chromosomes for the first time discovered by E.J. Balbiani in the year of 1881, and its significance for the first time were described by the painter in the year of 1930. So where these giant chromosomes are found? So generally the giant chromosomes are found in special tissues of some organisms. So generally these giant chromosomes are found in both plants as well as the animals. So in animals these were found in the diphterans. So these were found in the diphterans. So what are diphterans? The diphterans are nothing but insects with a pair of wing. Insects with a pair of wing. A single pair of wings are known as diphterans. So in diphterans, which tissues these are found? These are found in first one, salivary gland of diphterans larvae. Salivary glands of diphterans larvae. And second, these are also found in gut epithelial tissue. Gut epithelial tissue. These are also found in gut epithelial tissue, and these are also found in Malpighian tubules. Malpighian tubules. So what are Malpighian tubules? These are the excretory organs. These are the excretory organs in diphterans, similar to the kidneys in human beings. Next, coming to the fourth point, these are also found in the fat bodies. Fat bodies. What are fat bodies? The tissue which store fat. Tissue which store fat. So these are In diphterans, we can find these giant chromosomes in the salivary glands of the larvae and gut of the epithelial tissue and also the Morphegean tubules and also the fat bodies. So, in plants, where we can find these giant chromosomes. So, in plants, coming to in plants, where we can find these giant chromosomes. In plants, these giant chromosomes can be found in suspensor cell. Suspensors we can found in the suspensors are nothing but generally these are the small tissue which push the embryo towards the nutrient cell. This is the small tissue that push the embryo towards the nutrient cell so that the embryo should get the enough amount of nutrient to grow. So these are the suspensors. So this is the small tissue which pushes the embryo towards nutrients towards nutrients. So what are the distinct feature of giant chromosome? So let us discuss about these distinct features. First one. So these giant chromosomes are extremely or abnormally larger in size, larger in size. Second one. The second distinct feature of these giant chromosome is somatic pairing. Somatic pairing. And third distinct feature is formation of formation of dark and light bands. Dark and light bands. And the fourth distinct feature is formation of puffs. Formation of puffs. So let us discuss one by one these distinct feature in order. So first one, extremely larger in size. So first distinct feature is, so the first distinct feature is extremely larger in size. Extremely larger in size. So did you think why these giant chromosomes are heavy and extremely larger in size while compared to the normal chromosome? Because there is a feature in this giant chromosome known as endoreduplication. There is a feature in this giant chromosome that is known as endoreduplication. Very important for the examination. So that is endoreduplication. Endoreduplication. What is endoreduplication? So these chromosome will never enter into the M phase, that is the cell divisional phase. So they will always will be in the interphase. They will not go into the M phase, that is the cell divisional phase. So continuously they will be repeatedly going on into the interphase. The size of these chromosome will become large. That is, for example, let us consider endoreduplication. Let us consider a chromosome. Let us consider a chromosome which undergo endoreduplication. For example, first one in S phase, in first S phase, that is first interphase, two chromatids will form. In second, again four chromatids will form. In third, again 8 chromatids will form. In fourth, again 16 chromatids will form. Like this continuously, the chromatid numbers of these chromosome, the chromatid number of these chromosome will be double. chromatid number will be double for every interface. That is, in interface we will see G1, S phase, G2 phase. So in general these chromosomes will not enter into the M phase. They will only rotating on like in the interface. They will go to G1 phase, S phase, G2 phase, again G1 phase, S phase, G2 phase. Like this they continuously moving in only the interface. Hence as a result there is no proper cell division. So the chromatin number will get continuously increase, going on, going on, going on. So finally there become more number of chromatids and extremely larger in size. So this phenomena is known as endoreduplication. What is endoreduplication? The cell does not enter into the M phase. The cell do not enter. The cell do not enter into the M phase. Do not enter into the M phase. This is known as endoreduplication. Hence, as a result of this endoreduplication, these chromosomes are very larger in size. As the chromatin numbers will be going on increasing, increasing, increasing. As a result of this, the chromosome will become very larger. So this is the first distinctive feature of this special chromosome, that is this giant chromosome. Next, coming to the second point. So coming to the second point, that is somatic pairing. Somatic pairing. So what is somatic pairing? Generally, while compared to the normal chromosome, so when the normal chromosome will pair, generally here we'll discuss about normal chromosome, and here we'll discuss about our giant chromosome. So when the pairing will start in the normal chromosome, in the zygote stage of prophase 1 of meiosis 1, zygote stage of prophase 1 of meiosis 1. Here pairing will takes place for the normal chromosome. But in giant chromosome, the homologous chromosomes are the homologous chromosomes are always remain in pair. Always remain in pair. They won't get separated. The giant chromosome, the homologous chromosomes are always remain in pair and they won't get separated as that of the normal chromosome. Generally, in normal chromosome, what will happen in prophase 1 of the in zygote, the homologous chromosome will become pair. After that, in diplotin, they will become far away from each other. So pairing and depairing will be takes place. But in the giant chromosome, no such phenomena will happen. No such thing will be happen. So only in giant chromosomes, the homologous chromosome which are formed are always in the paired. They always remain in the paired form. So these chromosomes are always in paired. So as a result of this, what will happen? As the result, as a result, what will happen? The total number of giant chromosome, the total number of giant chromosome, the total number of giant chromosome is equals to haploid number of an organism, is equals to n number of an organism. For example, so for example, Drosophila. So if anyone asks how many giant chromosomes are present in Drosophila, so its haploid number, what is it? 2n number. 2n is equals to 8. So n is equals to 4. So there are totally 4 number of giant chromosomes are present. Hope you understand how this the total number of giant chromosome is equals to its haploid number because of this somatic pairing. In giant chromosome, the homologous chromosome will remain always pair, or they remain always attached together. Hence, the total number of giant chromosome is equals to its haploid number. Next, the coming to the third distinctive feature, that is formation of, that is formation of darkened light bands. Darkened light bands. Formation of darkened light bands. So generally, the giant chromosomes are generally very larger in size. So it can be easily appear in the compound microscope. So when scientists tried by staining it and they observed in the compound microscope, they observed the darkened light band patterns on the chromatid of these giant chromosomes. So they observed the darkened light patterns. So, for example, when these chromosome, that is these giant chromosome were stained and observed in the compound microscope, so they will appear like this. They appear like this. For example, this is one of the chromatid of this giant chromosome. Let us assume. So, this is the dark band. Alternate dark and light bands are formed. Alternate dark and light bands are formed like this. Alternate light and dark bands are formed. So, this is the dark band, this is the dark band, and this is the light band. Light band, it is also known as interband. Interband region. It is a interband region because it is present in between two dark bands. Hence, it is called as interband or light band. So, what happening in this interband and light band? So, that is interband or light band or dark band. So, what is happening? So, here the genes which are present in this dark band, that is the genes which are present in the dark band are densely coiled or densely packed that is the DNA is highly coiled. DNA is highly coiled. DNA is highly coiled in this dark band. So what about light band? In light band, this DNA is loosely coiled. DNA is loosely coiled in light band. DNA is loosely coiled in light band. So, as a result, generally in this dark band, heterochromatin is present. Heterochromatin is present. Heterochromatin is present in this dark band, and in the light band, euchromatin is present. Euchromatin is present. So, the genes which are densely classed, already we are discussed in that Lambert's chromosome, when the DNA is highly packed, when the DNA is closely packed or in condensed state, the genes which are present in the condensed state of the DNA is not available for transcription. Similarly, here also same thing, the genes or the DNA which is highly or densely coiled in this dark band are completely inactive. The genes which are present in this dark band are completely inactive, and the genes which are present in this light band are active. So these are open for transcription. They are actively participate in transcription, whereas the genes which are present in this dark band cannot participate in transcription. This is the main reason. So let us let us write the difference here. So dark band and light band. Dark band and light band. So what is in dark band? Genes are packed tightly. Genes are packed tightly. Here genes are loosely arranged. Loosely arranged. Next one. Genes are inactive. Here genes are active. Here no transcription takes place. No transcription takes place. Here transcription takes place and synthesis mRNA. So this is the complete story of formation of dark and light bands. How these dark and light bands are formed because of these, the DNAs or the genetic material which are tightly packed or form as a dark band, and the genetic material which is loosely packed or which is in uncondensed state will be appear as this light band. So coming to the next distinctive feature, that is the important distinct feature, that is fourth one, formation of formation of puffs. Formation of puffs. This is the one of the important and most distinct feature of this giant chromosome. Formation of puffs. Let us discuss how these puffs are formed. So as we discussed earlier, like this. So let us draw the picture of this giant chromosome. So for example, assume this is a chromatid of a giant chromosome like this, and here are the dark bands. The dark bands are present here, and these are the sides are the light bands. So these are the dark bands. So this is the dark bands. So now how these puffs will be formed? So as we discussed, the genes are very tightly coiled here. The DNA is very tightly coiled. Genes are tightly packed here. Okay, and here the genes are very active. Genes are inactive in this dark phase. Now let us assume a cell. This cell need more amount of RNA, more amount of mRNA to perform its function. The cell need more amount of mRNA to perform its function. When it does not reach its requirement, what will happen? The cell trigger the genetic material. The cell trigger its genetic material. As a result, what will happen? So the DNA which is present in highly condensed state, here the DNA which is present in highly condensed state will slowly become decondensation. That is, the highly coiled DNA become uncoiling starts. The uncoiling of the DNA starts. That is, the DNA which is present in this band will slowly become loosen. That is, the slowly become loosen and form the structure like this. Slowly become loosen and form the structure like this. And from here also the DNA slowly become loosen and form the structure like this. Similarly, here this will be formed like this, and here also this will form like this. Now what happened here? The DNA which is present in the condensed state now become decondensed, and the genes which are tightly packed now became loosely packed, and now these genes are available for easy transcription. Now these genes are available for easy transcription. Now these genes are available for easy transcription. Now these genes will undergo transcription. As a result of transcription, mRNA will produce, and this mRNA is utilized by this cell. This is the mechanism how these puffs are formed, and these puffs are the important site of transcription. Hence, we call puffs are the puffs are the important site for important site for transcription. Important site for transcription. So this is the complete process how these puffs are formed and why these puffs are formed. When a cell need a more amount of mRNA, so the cell will trigger to this the genetic material. As a result of that mechanism, the DNA which is tightly coiled here will become slowly decoil, or uncoiling of the DNA takes place. As a result, the genes become loosened, and the loosened genes will active, so and participate in the transcription. Okay, this is how these puffs are formed and the mechanism of formation of the puffs. Here we can discuss the another point, that in Lamb-Rush chromosome, loops are the important site of transcription. Loops are active site of transcription. Active site of transcription. Transcription in Lamb-Rush chromosome. In Lamb-Rush chromosome, here in giant chromosome, puffs are the important site of transcriptions. Puffs are the important site of transcription. Transcription in giant chromosome. In giant chromosome. So sometimes these puffs are induced by, that is, the formation of puffs is induced by the hormone in some diphterans, that is the hormone known as ecdysone. The hormone known as ecdysone. The hormone, the hormone ecdysone, what it will do? It will induce the, it will induce the formation of puffs. It will induce the formation of puffs in the diphtheran giant chromosome. Diphtheran giant chromosome. Giant chromosome. What this hormone will do? Simply, so this is a molting hormone. This help in molting. This is a molting hormone. Molting hormone. So it need more amount of, for the molting, it need more amount of mRNA. As a result of that, it will trigger the formation of puffs in the dipterans of the giant chromosomes. In giant chromosomes, this will induce the formation of the puffs. So these are the 4 distinct features of these giant chromosomes. Next topic, coming to the structure and function of a giant chromosome. As we discussed, so now let us discuss a simple example of the structure and function of a Drosophila giant chromosome. The giant chromosome of a Drosophila. Giant chromosome of a Drosophila. So now let us discuss about the complete structure of a Drosophila giant chromosomes. So as we previously discussed, the total number of giant chromosome is equal to its haploid number. The total number of giant chromosomes. The total number of giant chromosomes is equals to haploid number. So in Drosophila, total number of giant chromosome is equals to 4. So now let us see the picture of the giant chromosome of Drosophila. So this is the picture of this giant chromosome of a Drosophila. So here we'll discuss about one point. So this is the region. So here you can observe at the center. Here at the center, this is the region known as chromocenter. This is known as chromocenter. Chromocenter, because here you can observe where all the centromere of the chromosomes, all the centromere of the chromosome meet at a one point. That point is known as chromocenter. So let us see the 4 chromosomes of the 4 giant chromosomes of the Drosophila. So here you can observe the one X chromosome of the Drosophila, and there is another X chromosome which is exactly present the below of this chromosome. So in picture we can't see the another chromosome, but there is another X chromosome which is present below here. So now here you can see the right arm of second chromosome. So this is the second chromosome. This is the right arm of the second chromosome. So this is the left arm of the chromosome 2. This is the left arm of the chromosome 2. That is, this chromosome will be like this. So we'll say, so this chromosome is like present like this. Like this. So this is chromosome number 2. This is chromosome number 2. This is the right arm and this is the left arm. So generally in BDC, they given the structure of this chromosome. So many students will get confused. So this is the exact picture of this Drosophila giant chromosome. So this is the second chromosome. So this is the left arm of the second chromosome, and this is the right arm of the second chromosome. Coming to the third chromosome. So this is the right arm of the third chromosome. This is the right arm of this chromosome number 3, and this is the left arm of the chromosome number 3. So let us draw how it is present. It is present like this structure. So like this structure, and this is the small chromosome, the small chromatid which here appearing is the chromosome number 4. This is the chromosome number 4. Chromosome number 4. So now there is also a Y chromosome present in it, which is, as a Y chromosome is very small, so it is embedded in this structure. It may be present here or somewhere, but it is a very small, so we can't see in the picture. So here you can see the formation of puffs. So this is the puff. This is the puff, which is an active site of transcription. This is the active site of transcription. So here like this you can see the puffs. So here also the puffs are present like this. The puffs are present like this, and they will appear as a dark band and light band in the chromosome when we stain. So this is the chromocenter. As we earlier discussed, this is the chromocenter. So as a result, the centromere, see, this is the centromere of chromosome number 3, this is the centromere of chromosome number 2, and also the X chromosome number. So all the centromeres will meet at one point, and this point is known as chromocenter. This is known as chromocenter. So as a result, many chromatids, see, many centromeres, many centromeres are meeting together, are meeting together at a point, at a point, right? Hence these are also known as polytene chromosomes. These are also known as polytene chromosomes. Polytene chromosomes. So poly means many. So poly means many, and teen means thread. So teen means thread. So many thread-like structure, that is chromatids, all are meeting together at a point, hence these are known as polytene chromosome, and the condition is known as polyteny. So the condition is known as polyteny. So this is the exact structure of these giant chromosome, 4 giant chromosomes of the Drosophila. So here you can see the dark band, interband, and the dark band when it form the puff. When it form the puff, it appear like this. So generally the puffs are only arised from the dark band. So you want to remember one point, that here puffs are, so puffs are arising from, puffs are only arising from dark band. These are only arising from dark band. So here in this picture you can clearly observe, and also you can clearly understand how the puffs are formed. Why the puffs are formed? Why the puffs are formed? Because if a cell need more amount of RNA, so in order to synthesize, in order to meet the cell requirement, the puffs are formed. So this is the complete topic of the special chromosome. The in the first topic, that is in previous class, we discussed about the Lambert chromosome. Now we discussed about the giant chromosome. These are the two different types of uh, these are special chromosome, and in there are two other special chromosome, that is the nucleus of prokaryotes, and as well as the accessory chromosomes or B chromosomes. So generally these two chromosomes are not considered that much very important, and they never ever ask in exam. So for the exam point of view, you should prepare only this Lambert chromosome and the giant chromosomes. So now we'll do a brief about we'll write a difference and similarities between the Lambert's chromosome and the giant chromosomes. So differences and similarities we'll write in one table. For example, first one, Lambert's chromosome. So Lambert's chromosome and giant chromosomes. Giant chromosomes. So first one, in Lambert's chromosome. So in Lambert's chromosome, what will happen? Loops are formed. Here loops are formed. Here puffs are formed. Puffs are formed. So the loops are the active site of transcription. Loops are active site of transcription. Active site of transcription, and here the puffs are the active sites of transcription. Active site of transcription. Here no interbands are formed, that is the dark band and light bands are not formed in the Lambert's chromosome. No interbands are formed. No interbands are formed. Here interbands are formed. Interbands are formed here. Interbands are formed here. So these can be easily observed in compound microscope. Easily seen in compound microscope. So these also easily observed in compound microscope. Compound microscope. Next, these are observed in first one, many invertebrates, all vertebrates except mammals, all vertebrates except mammals. In mammals, the two exception, that is humans and rodents. Humans and rodents exception. And these are also found in amphibians. Here these are found in tissues of diphtheria. Tissues of diphtheria. So here is the complete difference and the similarity between the Lambert's chromosome as well as the giant chromosome. So finally the topic is completed, and the very simple topic, both the special chromosomes are very important for the exam point of view. So don't miss this topic. So generally you should not no need of that more information. So main point you should remember for the exam point of view. So finally, thank you for watching our video. So if you like this video, please share, subscribe and follow to Agrivyas Academy YouTube channel for further more updates. Please follow and support us. Thank you.